Question No. 1. Define antigen and antibody. What is the difference between active and passive immunity? Answer. Antigen. The term originally came from antibody generator. An antigen is a substance or molecule that when introduced into the body, triggers the production of an antibody by the immune system. Antibody then kills or neutralizes the antigen that is recognized as a foreign and potentially harmful invader. Antigens are usually proteins or polysaccharides. This includes parts, coats, capsules, cell walls and toxins of bacteria, viruses and other microorganisms. Antibody. An antibody is also known as an immunoglobulin. It is a large Y-shaped protein used by the immune system to identify and neutralize foreign objects like bacteria and viruses. The antibody recognizes a unique part of the foreign target, termed an antigen. Antibodies are produced by a white blood cell. Example: IgA, IgD, IgA, IgG and IgM. Active immunity. Antigen activates B cell or T cell of the individual. Antibodies or effector T cells are produced by the individual in infection or by vaccination. Immunity develops in 1 to 3 weeks. Immunological memory develops. So, immunity may persist for years to lifelong. Active immunity used to prevent the disease. Hypersensitivity does not develop. Passive immunity. Performed antibodies are passively obtained or administered. B cell or T cell of the individual is not involved as no activation by antigen. Individual does not produce antibody or effector T cell. Immediately available. Effective means immediate. Last for few weeks, no memory. Used for treatment or for prophylaxis. Hypersensitivity may develop. Question No. 2. What is immunization and vaccine? Describe types of vaccine with example. Answer. Immunization. Immunization is the process whereby a person is made immune or resistant to an infectious disease, typically by the administration of a vaccine. Vaccines stimulate the body's own immune system to protect the person against subsequent infection or disease. An individual may be immunized by A. Active immunization, vaccination. B. Passive immunization, protective antibodies are used in serum. Active immunization or vaccination. Vaccinations are done with vaccines in which antigens are introduced. So, vaccine are used for artificial active acquired immunization. Passive immunization, performed antibodies are artificially introduced in a host. Temporary protection against infection can be achieved. Serum preparation are used for passive artificially acquired immunization. Vaccine. Vaccine. A vaccine is a preparation that improves immunity to a particular disease. A vaccine typically contains an agent that resembles a disease-causing microorganism and is often made from weakened or killed forms of the microbe or its toxins. The agent stimulates the body's immune system to recognize the agent as foreign, destroy it and remember it, so that the immune system can more easily recognize and destroy any of these microorganisms that it later invade. Types of vaccines. 1. Inactivated or killed vaccine. Inactivated vaccines are composed of microorganisms that have been killed with chemicals and/or heat and are no longer infectious. Examples are influenza vaccine, rabies vaccine and cholera vaccine, bubonic plague vaccine, polio vaccine, hepatitis vaccine. 2. Live attenuated vaccine. Live attenuated vaccines are composed of microorganisms that have been cultivated under conditions which disable their ability to induce disease. Examples are yellow fever, measles, rubella, mumps, typhoid, 3. Toxoid vaccine. Toxoid vaccines are made from inactivated toxic compounds that cause illness rather than the microorganism. Examples of toxoid-based vaccines are tetanus, diphtheria. 4. Subunit vaccine. Subunit vaccines are composed of small fragments, protein, rather than introducing an inactivated or attenuated microorganism to an immune system, which would constitute a whole-agent vaccine. This fragment can create an immune response. Example: hepatitis B vaccine is a subunit vaccine that is composed of only the surface proteins of the virus. 5. Conjugate vaccine. Certain bacteria have polysaccharide outer coats that are poorly immunogenic. By linking these outer coats to proteins, e.g. toxins, the immune system can be led to recognize the polysaccharide as if it were a protein antigen. This approach is used in Haemophilus influenza type B vaccine.